Hello bachcho. Let's see the question. So the question says a piece of nucleic acid used to find out a gene by forming a hybrid with it is called as. So इन चार ऑप्शन में से हमें वो न्यूक्लिक एसिड को आइडेंटिफाई करना है जो एक जीन के साथ हाइब्रिड बन सकता है एज वेल एज उस जीन को आइडेंटिफाई करने में हेल्प करेगा. So the key concept in this question is to understand gene identification or gene detection techniques. So, with the help of a technique known as nucleic acid hybridization, we can identify or detect the presence of a particular sequence of DNA or a gene. So, in this technique, we require a probe. So with the help of a probe, we can detect the presence of a particular gene in a sample. So this probe is actually a short fragment of DNA or RNA. Now, this short fragment of a nucleic acid is actually labeled. So now this molecule can either be radioactively labeled or it can be labeled with the help of fluorescence. Also, this probe is sequence specific. That is, this probe has the complementary sequence to the target DNA. So the gene that we want to identify, so it has a particular nucleic acid sequence. That gene will have a particular nucleic acid sequence. And this sequence is known as the target DNA sequence. Now the probe will have a sequence which is complementary to that target DNA sequence. Now this complementarity is necessary so that the probe can hybridize with the target DNA sequence. So that is what is nucleic acid hybridization. So if we see the steps of nucleic acid hybridization, the very basic steps. So firstly, we have the DNA sample. So this is the long nucleotide sequence of DNA sample. So firstly, we are going to denature this DNA sample and then we'll get the two single-stranded DNA strands. Now in that single-stranded DNA strand, we are going to have a gene of our interest. So consider if this is the nucleotide sequence which is the gene of our interest. So now this will be known as the target DNA and this target DNA has a particular nucleic acid sequence. Now the DNA probes which are designed will be complementary to the target DNA. So we can say that the DNA probes are complementary to the target DNA. Also, these probes are labeled either radioactively or with the help of a fluorescence. So this molecular beacon is actually the labeling molecule which is attached to the probe. Now, with the help of nucleic acid hybridization method, these molecular probes will identify the gene of interest, that is a target DNA, because it is complementary to that sequence and it will result in the binding of the probe with the gene of our interest or the target DNA. So this is the DNA hybrid which is formed between the target DNA and the probe molecule. due to the hybridization or due to the presence of complementary sequence in the probe molecule, which then identifies the gene sequence and then binds or hybridizes with the target DNA. So this is the basic steps of nucleic acid hybridization, and with this we can identify the presence of a particular gene. Now, this probe or the DNA hybrid which is formed can be identified with the process of autoradiography. So, with the help of autoradiography, we can identify whether the probe has bound to the target DNA. So, if the probe has bound to the target DNA, then we will observe fluorescence or radioactivity depending upon the type of labeling molecule that is utilized. So, if the gene of interest or the target DNA is present in the DNA sample, then the probe will hybridize with that DNA sample, and this will show fluorescence or radioactivity in the autoradiograph because the DNA hybrid has been formed. And if the gene of interest is absent in the sample, then the probe will not bind to the target sequence, and therefore the fluorescence or radioactivity will not be observed in the autoradiograph. So this is the basic technique of nucleic acid hybridization. So now if we come back to the question says that the piece of nucleic acid which will be used to find out a gene. So we are finding out the gene with the help of a nucleic acid hybridization method, and this piece of nucleic acid is forming a hybrid. So we saw that probe is the molecule which forms the hybrid with the nucleic acid sequence. So this DNA probe will be the final answer. So option 2 will be the right answer because DNA probe is the one that is used to identify a gene with the help of nucleic acid hybridization by forming a hybrid with the target sequence. Now, cDNA will not be our answer because complementary DNA is utilized when RNA is converted into DNA. Sticky end is formed when restriction endonucleases uh, results in the cutting of the DNA at a particular sequence. So again, sticky end is not used to form a hybrid. Also, blunt end is also formed when a restriction endonuclease cuts the DNA at a specific position. So again, a blunt end is also not a one which is used to identify the presence of a gene. So our final answer is option 2, that is DNA probe. Hope you understood this well. Best of luck. Thank you.